Um, Dario, you 5 years ago won a very major prize awarded by the Institut de France, the Prix Le Foulon de la Lande, for your discovery of the biological target that is responsible for the fact that a new drug has been developed to treat heart disease, ivabradine. Can you first of all say, how did that happen? How did you end up discovering and working on what became the target for a major pharmaceutical development?
Yeah, sure. This is actually one of the rare events where you can link a basic finding to a practical one. When we discovered, and that was a long time ago in the late 70s, that happened in Oxford, of course, this mechanism, we were very surprised because this was a very strange mechanism with properties unknown before, and we actually, this is the reason why we call this mechanism Fanny current. And I'm talking about an ionic current, and so what flows across ion channels. Ion channels are membrane components, proteins, which allow ions going across and change the electrical activity of cells. And there are, of course, very many ion channels, each of which has a specific function. So when we discovered this thing, we were very, very puzzled because it didn't have any known properties or properties from the known channels at that time. And but at the same time, as much as being puzzled, we realized soon enough that this component could have interesting properties in terms of generating spontaneous activity. We were very, very excited because of course we had recorded this component from pacing tissue, pacemaker of the heart, and we realized very soon that not only was this component able to generate spontaneous activity, but it was also able to modulate cardiac rate, a fantastic property which led us to define the current as pacemaker current as well as Fanny current. And indeed, that was the original finding, and then we went on and on to characterize this component. And as a matter of fact, to make a long story short, what this component does is to change the electrical activity of cells in such a way that these cells are essentially spontaneously active. They don't need any external trigger to contract, and they are localized in a very special region of the heart, which is the pacemaker, the natural pacemaker of the heart. This very special property belongs to the cell themselves, so that you can isolate these cells and they beat spontaneously for whichever time they are allowed to do this. This component, therefore, is able to determine the rate at which spontaneous activity occurs. And of course, what's the importance there is that this rate is not only the rate of the single cells or the region of cells with this property, it is the heart rate, because these cells actually give the stimulus for the whole of the heart activity. So beating rate is determined by the properties of these cells and actually by the properties of this current. What happens is that essentially the amount of current that goes across these channels determines the frequency of the heart. So you can change the rate of the heart by simply modifying the properties of these channels, and particularly by increasing or decreasing the number of channels. If you increase the number of channels, then you will have an acceleration of rhythm. If you decrease the number of channels that are active, that participate, then the frequency will be slowed down. And this is actually exactly what happens when the heart is subject to the stimulus generated by, for example, the sympathetic autonomic system. So adrenaline accelerates the heart through this component, at least in an important way. This is not the only mechanism that occurs, but in an essential way. And also the parasympathetic, the vagal stimulation, decreases heart rate through this component by decreasing the number of channels. So obviously this opened up a lot of possible interventions and applications, one of which was really successful, and was the idea is relatively simple. If you find a drug which binds and blocks or reduces, inhibits the activity of these channels specifically, and this can be done with the instrumentation and the techniques that are available today, then you can control heart rate from just using a specific drug. And this is what happened. This is the finding. This is what led to the finding of this new drug. The new drug whose action is that of binding specifically to these channels, and of course this leads to slowing of heart rate. A fantastic thing from the point of view of the pharmacological interventions, because there are many pathologies which are take advantage, have benefit from slowing of heart rate. For example, people with angina. Angina is a very painful pathology linked to cardiac disease, and whenever the best way to reduce the angina attacks is to reduce the heart rate, because the amount of oxygen that the heart uses depends in a proportional way with its rate. So if you decrease the rate, you slow the heart down, then the amount of oxygen that you have to provide to the heart itself is lowered, and this is a fantastic way to inhibit the pain, particularly for the coronary heart disease patients. And so this actually, the finding of this and the synthesis of this drug led to a very important step forward in not only in underlining, stressing the physiological relevance of the current in terms of generation and control of heart rate, but also from the point of view of clinical application.
One further question, Dario. That's an excellent description of the physiology and then the development of the drug. How long did it take from the initial discovery of this mechanism to the development of a usable drug?
Yes, it took a long time, as a matter of fact, because this system was found in 1978 and then published in 1979, and then only in 2005, 2006. So we needed some 25 years or so, and more than that. And that was obviously necessary from several points of view. First of all, because although the finding was well received by the scientific community, it penetrates it penetrated the scientific community only with a relatively short speed, but in the end, I think that this convinced drug companies to search for that kind of drug specifically. I actually remember that some of my colleagues, when the first of these components, because many components were developed before the development of ivermectin. I remember one of my colleagues was given a contract with the drug company to determine the action of a drug on heart rate, and the work done by this colleague and his laboratory led to the description of this drug as a calcium calcium blocking agent, calcium channel blocking agent. Such was the determination and the lack of confidence in finding channels. And only when the same drug company came to my laboratory were they able to understand and to discover and to find that this drug actually was a finding channel blocker. So there was some resistance in the scientific community that led to a sort of delay. But in the end, I think that what has been found is really very important from the application point of view.
One final question, Dario. Do we know how many patients have now been treated with ivermectin? Must be thousands and thousands, mustn't it?
I don't have the exact number, of course. One has to ask for the various clinics and hospitals, but it's a very large amount. It's now being used in very many, I don't know how many countries. And recently, I understand from the people working for the drug company who developed this drug, that the drug has been approved also in the United States. That was a little bit of market that was missing, but now it has been developed and it has been commercialized also in the United States, and they expect a very good market sale there.
Well, thank you very much for telling us about the story, Dario. A fascinating move from all the way from some fundamental research about 33 or 4 years ago, all the way through to the development of a new medication. It's a fantastic story. Thank you very much.
Thank you.